Well, thank you so much. I will remember this moment for forever. And there are a lot of people to thank, including David Ellis, the president of the university, but also the director of the Institute of Genetics and Development, as well as the dean of the Life Sciences Department. I also want to thank the research community at the University of Rennes, as well as my collaborator and my great friend, Professor Francis Galibert. I am sure that there are many people who are equally qualified to be standing here, and I am both grateful and humbled by this recognition. It's particularly gratifying to visit here and have a chance to talk to some of my previous and current students, postdoc fellows, and colleagues, because none of the achievements reflected in this award took place in isolation. Indeed, our work summarizes the collegiality and the generosity of countless scientists with whom we have worked for decades. To begin, let me just tell you a couple of things about myself. Early in my career, I couldn't decide whether I wanted to do human or canine genetics, so I pursued both. Together with Professors Kathleen Malone and Janet Daling, as well as my own postdoc fellow, Dr. Amy Langston, we were the first to study the frequency and the distribution of mutations in the BRCA1 and BRCA2 genes in young women drawn from the general population. We showed that many more women carry these genes than was previously suspected, with up to 87% of those going on to get breast cancer and 45% to get ovarian cancer. 3 years later, a phone call from Dr. Janet Stanford in Seattle, Washington, pulled me into prostate cancer genetics, which has proven very challenging to unravel. Over 50 genes contribute to a man's risk for getting prostate cancer. And those genes account in part for the very high risk of disease that's observed in some families. In the families in this slide, each man colored in black has prostate cancer. So these are truly remarkable families. Janet and I have studied 300 of such families, particularly those with aggressive disease, in an effort to find predisposing mutations. We have found over 30 such variants. And we've also shown that some genes initiate breast or prostate cancer by very subtly destabilizing DNA. And we've also shown that many people, perhaps some in this audience, carry mutations in these genes without ever knowing it. Now, the other half of my laboratory worked on building maps to navigate the canine genome. We quickly realized that doing this alone was not a good idea, and led by Professor Galibert, together with Professors Greg Ackland and Matthew Breen from the United States, we developed high-density maps of the dog genome. 2 of the dog's 38 chromosomes are shown here, and don't worry about the details. But know that maps like these allowed researchers to navigate up and down each canine chromosome, knowing precisely where they were, as well as back and forth between the dog and the human genome. This, in turn, facilitated the identification of many disease genes, including those for cancer, heart disease, deafness, and blindness. As it became clear that the dog offered a host of opportunities for studying not only disease, but also the genetics of morphologic variation and behavior, dozens and dozens of scientists joined us, producing papers that linked issues such as domestication of dogs from wolves, the development of breed with genes that are important in development, all together. Our own papers showed that major morphologic traits in dog breeds, such as body size, skull shape, and limb length, are all controlled by a very small number of genes, each of which has a very large effect. And this was a major milestone for us, and it really highlighted the value of the dog system for studying what are very complex traits in humans. Further, finding that the same genes, when just slightly modified, cause major diseases in humans, such as cancer and obesity and birth defects, was very surprising, and it was very exciting for our field and many others. For instance, this is one of my favorite pictures. We identified a series of genes that account, in large part, between being a 200-pound Great Dane versus a 2-pound Chihuahua. Now remember, all dog breeds are members of the same species, and they can be crossed to produce fertile offspring despite their size. So if you want to do this cross, I would think carefully before. I would plan it out very well. Now, we found that several genes contribute to giantism in dog. One called ACSL4 makes big dogs really big. But the same gene, because it plays a role in lipid biosynthesis and fatty acid degradation, when mutated in humans, causes insulin resistance, effectively starving cells of nutrition. Another gene, IGF-1, also helps make big dogs big, but it does so through the thyroid pathway. And this gene, when very subtly mutated in humans, causes something called metabolic syndrome, which in turn increases the risk of heart disease, stroke, and diabetes. These are all nice examples of why the Dog Genome Project has to exist. By studying the genetics of breed variation, we enlighten our understanding of critical human disorders. All of the research efforts and so many more that I've talked about were led by trainees. I've had many wonderful graduate students and postdocs over the last 25 years, and I truly apologize for not naming each. But you should know that my trainees, including those from Rennes, frequently argue with me, and they find it very sporting to try and prove me wrong. Often they are successful, but don't tell them that. One story that comes to mind is that of a young technician named Dana Mosier, who, despite my instructions not to do a particular experiment, because I thought it had no point and would never work, went ahead and did it anyway. She genetically mapped a set of whippets that the owners had been complaining about because they were bulky and very heavily muscled and not long and lean like a typical whippet. She found that a single base pair change in a gene called myostatin dramatically increased muscle mass. Now, the dogs in the first column are normal. The dogs in the second column have one copy of the mutation, and you can see that they're much more muscular. And the dogs in the third column have two copies of the mutation, and you can see that they're extremely muscular. This turned out to be a major finding, and the paper not only ended up on the cover of a prestigious journal, but it was reprinted several months later as one of the best papers published that year. I had to apologize to Dana. Oh, and now I call her Professor Dana Mosier. So this work was the beginning of our ongoing efforts to understand how early selection by humans resulted in distinct occupations among dog breeds that are absolutely critical for human survival. Without breeds having the right physiology and neurologic connections to herd, to hunt, and to guard, humans as a species would not exist today. Now, our most recent work has focused on completing the whole genome sequence for 10,000 canines in an effort to develop a resource for everybody in our community. As a start, led by Dr. Joss Placé, who trained here in Rennes, we have determined the whole genome sequence for 722 canines. That is, we have found every gene, every base pair, and identified 91 million variants in the DNA sequence amongst those 144 breeds that make up this study. And this work, in turn, has formed the basis of our canine cancer studies. You should know that cancer is a major disorder for dogs. Just like humans, 1 in 4 will get it at some point in their life. We've been interested specifically in invasive bladder cancer, showing that the dog is an outstanding model for human bladder cancer due to a similar age of onset, very similar pathology, response to treatment, and site of metastases. We've also shown that a gene called BRAF is critical for tumor growth, and that this gene represents a target that can be used for treatment. Further, we show that the disease can be now diagnosed by a very simple urine test rather than invasive and expensive imaging. Our interest extends to several other types of cancer, including histiocytic sarcoma, which we study with Catherine Andre and Benoit Idan in a long-term collaboration here at the University and the Institute of Genetics and Development in Rennes. We also work on genetic susceptibility to gastric cancer and squamous cell carcinoma. And we recently showed that upregulation of a gene called KIT ligand is critical for the development of carcinoma. The upregulation is caused by increasing production of the protein via an enhancer sequence that's present in variable number of copies. The more copies you have, the more protein is produced, and the greater the risk for cancer. Now, all of the trainees who led these studies share the vision, the dedication, and the fortitude to be great scientists. Indeed, it has been my honor to witness their scientific achievements as they matured from trainees to becoming my colleagues. But I would single out a few of those in particular who I worked closely with and who trained with Professor Galabert here in Rennes, who then served as postdoc fellows or students in my lab. And this includes Pascal Quignon, Edward Cadu, Maud Rimbaud, and Jos Places. We've also collaborated with many people in the Galabert group, but Christophe Hyatt, Catherine Andre, Thomas Derrien, and Benoit Idan have worked with us for years and years, and we are extremely grateful to them for their collaboration and generosity. D'un point de vue personnel, au cours de mes nombreuses visites en France, j'ai toujours eu plaisir à apprendre davantage sur sa culture et ses habitants. Ma famille et moi-même partageons la même tristesse avec Paris, avec la France, mais aussi celle du monde entier concernant les ravages de Notre-Dame. Nous vous remercions pour tout ce que vous avez sauvé, mais nous sommes tristes pour ce qui aura été perdu. My thank yous would not be complete without some acknowledgement of my family. They have been my greatest source of inspiration and motivation. Before my father died 4 years ago, he gave me 3 pieces of advice, which I would share with you today. He told me first to cherish my family and my friends, and to understand that each would write their life story, and that they should be able to do so without my criticism and with my support. I try to do this for my students every day. He told me to stay humble and to understand that always there would be greater and lesser, smarter and duller, and braver and more foolhardy persons than myself. He's right, and I've learned from all of these people. Finally, he said, "Dear God, don't marry a bum like your cousin did." I like to think I achieved at least the last of these goals, and that's why I thank my husband of 25 years, who unfortunately could not be here today. The world knows him as a brilliant neurobiologist, I know him as a patient husband, a compassionate father, and someone who supports my science and my trips to Rennes unconditionally. In closing, I would tell you I'm frequently asked what I would do with my life if I were not a scientist. In truth, I actually knew from the time I was 14 that I wanted to be a geneticist. I know now that as a scientist, I am exactly where I was meant to be in life. When it is late at night and I'm alone and I'm staring at data I've looked at for months or even years, and I observe something that I missed 100 times before, it's a rare and precious moment. It's truly a remarkable thing. For just a few moments, I am totally alone in my understanding of something in biology, and I and the universe share a very private thought. Out of such moments come new ideas and inspirations, and I've dedicated my life to seeking such moments. My journey has been one of privilege, taken with colleagues, trainees, and family. On behalf of all of these people, I thank you most heartily for this prestigious award.
Thank you very much, Mrs. Ostrander, for this very moving and full of enthusiasm to talk.